chemicals released by damaged cells. Okay? Very aggressive phagocytic cells. And you can just see those little granules of potent chemicals in there. Okay? And so when they phagocytize a foreign invader, these little granules that contain digestive enzymes are released. The foreign invader is packaged up in a little vesicle itself, okay? And the vesicle fuses with these granules containing really nasty digestive enzymes that digest the foreign invader and kill it. Unfortunately, the enzymes also kill the neutrophil. Pus consists of dead neutrophils, so you should know that too. And that these are the most common, very hostile, aggressive phagocytic cells, first at the site of an infection. And there's a picture of a cell phagocytizing another one, and I'm not sure which one is the phagocytor and which one is the phagocytee here. Kind of hard to tell, but maybe that one is phagocytizing this one, it kind of looks like. So here is the epidermis, and one thing we said about the dermis, it comprised of loose areolar connective tissue that contained lots of immune cells. And one of the type of immune cells that migrates to the site via capillaries is neutrophils. So damaged cells spew forth their contents, and it is those chemical contents. Somehow the neutrophils can smell them a mile away, and maybe some of them get into the blood. And anyway, the neutrophils migrate via capillaries and actually ooze through the walls of capillaries in a process called chemotaxis. Is where neutrophils are attracted to the site of an infection and they phagocytize the foreign invaders. And if it's a significant infection, a bunch of dead neutrophils will accumulate, which can be visibly seen as pus. Okay. Eosinophils are the least prevalent leukocyte. They are effective against parasitic worms, and they release enzymes which digest attached roundworms in the intestine, most commonly. Basophils are cells we'll talk about again and again because when they degranulate, they release histamine. Histamine is the chemical mediator of the inflammatory response. These cells are primary players in acute allergic responses for people that are having allergies, which can be life-threatening. It is the acuteness of the inflammatory response that winds up killing the patient or causing intense misery to the patient. And we'll be talking about that this quarter. Okay. So this is where histamine comes from. Okay. And mast cells, basophils are in the blood. Mast cells, they can evolve into mast cells which migrate into the tissue. And it's really the same cell. A mast cell is really just a basophil that has migrated into the tissue. So you find them both in the tissue. And when I say in the tissue, I mean in loose areolar connective tissue. Because you remember loose areolar connective tissue has enough ground substance that they can move around in there, that these immune cells see. And so you find them in loose areolar connective tissue, which underlies all epithelial tissue. Okay, so that was granulocytes. Okay, neutrophils, eosinophils, and basophils. Phil means to like, and those cells get their names from the stains that are used to identify them. Neutrophil is stained by a neutral stain. that is neither acidic nor alkaline. Eosinophils are stained by an eosin, that dark blue eosin stain. Basophils are attracted to a basic or alkaline stain. So they're just named for the stains that identify them. The two types of agranulocytes are lymphocytes, and there are two types of lymphocytes, T cells and B cells. Cells of our immune system for the specific immune response that we'll talk about in excruciating detail in a couple weeks. Very, very interesting stuff. Monocytes are the largest white blood cells. They're most famous for the fact that they migrate into the tissues and evolve into macrophages. And macrophages are very important cells in the tissues, and of course we mean in loose areolar connective tissue. Although you find macrophages in lymph nodes and the spleen and all kinds of places. Mononucleosis affects monocytes. So there's a little synopsis, good little study thing to know a little bit about each of the different kinds of cells. So that's the cells. Some pathological conditions. You can recognize some of the words, like if you have your blood work done and you get the computer red flags you, and then there will be a little bit of writing and it'll just say erythrocytosis, which is very uncommon to see next to your name. That means you actually have too many red blood cells. Erythrocytopenia means you don't have enough red blood cells. Leukocytopenia means you don't have enough white blood cells. And leukocytosis means you have an abnormally high white blood cell count, probably have an infection somewhere. Okay. Mononucleosis, said to be self-limiting. I've known people that have claimed to have had it for years. So that's kind of, I haven't known anyone who had that. Seems to be a disease of younger folks for some reason. I don't know, it's just my observation. Caused by the Epstein-Barr virus, which is kind of a notorious virus, claimed to be the culprit in lots of different things. Lots of different kinds of leukemia. Maybe the most famous one is caused by the so-called Philadelphia chromosome, which is during cell division of a red blood cell, of these stem cells, the chromosomes aren't properly pulled apart, and two chromosomes get stuck together in a way that results in one big chromosome called the Philadelphia chromosome, and you wind up with a gene for white blood cell production is put right next door to a regulatory gene that makes that it turns on this white cell production gene to the point where it's producing way too many white blood cells. These two incorrect genes get stuck next to each other. The regulatory gene overregulates a white blood cell gene. That's just one kind of, that's actually curable to some degree. But there are all kinds of nasty leukemias. And literally, leukemia means white blood. So lots of different types of leukemia. Not a nice thing to be diagnosed with, though. Sickle cell anemia we talked about. Pretty easy to identify. An intriguing one, sleeping sickness, is caused by a protozoan, the trypanosome, which is carried by tsetse flies in places in Africa. And whole parts of Africa have been shut down just because, and rendered uninhabitable because of the presence of these plastic tsetse flies, which cause this sleeping sickness, which basically just kills everybody inside. And so, and there's, last I heard, there was no cure, definitely no cure, and no drug, or it's hard to prevent protozoan diseases. You can, like with amoebic dysentery, it's caused by an amoeba. There are drugs that can kill that. But the one that causes malaria, they still haven't found anything that's really effective against malaria, totally effective. They kind of like HIV, they treat the symptoms, but
So the vaccination?
there's no vaccine for malaria. And they're working on it. So, last I heard, they were working on a vaccine and weren't there yet. So, maybe you know something I don't, but, they have drugs for treating the symptoms, but it's just a protozoan that affects red blood cells. But you see, it's hard to vaccinate against a protozoan. It's a one-cell organism. Bacteria and viruses are much easier. So that is the cells. What about the plasma? Plasma proteins will be big with us. They're the protein fraction in blood, and they're responsible for the blood osmotic pressure, because they're by far the most prevalent solute. And remember, the higher the solute concentration, the higher the osmotic pressure. And the osmotic pressure, and remember how water wants to flow through a semi-permeable membrane in the direction of more solute? So all the blood would run out of the capillaries if it weren't for these plasma proteins. The plasma proteins are too big to get out of the holes in the capillaries, so the plasma proteins stay in, and so that holds a lot of water in. A lot of water flows out in the capillaries, carrying fuel, nutrients, and oxygen, but not all the water. About half of it stays in there, luckily, otherwise all of our capillaries would solidify. And so, very important, these plasma proteins. Organic solutes like hormones and nutrients, inorganic solutes like ions. Okay, so plasma proteins, they're responsible for blood osmotic pressure, and there are a whole bunch of important ones. Albumin is there mainly for blood osmotic pressure, but we have clotting proteins. Our gamma globulin fraction is our antibodies, just the circulating antibodies that we have all the time. And albumins, besides giving us blood osmotic pressure, do help bind certain nonpolar molecules become reversibly bound to plasma proteins, and so you can give more of a drug. You can give you have a time release mechanism where you can give a much larger dose of a drug because most of it is bound to plasma proteins and it's reversibly released into the tissues bit by bit, sort of a time release thing that allows you to give a larger dose. Rather than taking a pill every half hour, you can take one, twice a day or something. So, okay, organic solutes, waste products, hormones, nutrients, inorganic solutes, and all of these are very important to us. Various ions, so-called minerals. Minerals are just ions. The vitamins and minerals, the minerals are inorganic ions. Okay, and so, let's move on and talk about plugging up the pipes if they get a hole in them. We've talked about the cells and the plasma. What now happens if you get a hole in the pipe? Okay, we're showing a little teensy hole here. And so we have a number of mechanisms that come into play, and by far the most important players are probably the platelets, because they release all kinds, when they sense that something's wrong with the pipe, they release all kinds of chemicals. They're just, they're very discriminating, and the walls of blood vessels are intended to be very smooth. Okay, when platelets sense a smooth surface, they remain happy and they don't release all kinds of chemicals. And so all of the vessels in our body are lined with simple squamous epithelium, which is very smooth, okay? It's called endothelium in connection with blood vessels, simple squamous epithelium. A blood capillary, which is what we're showing here, is just a single, all it is simple squamous epithelium with nothing else, okay? And so anyway, the stages of hemostasis, plugging the hole. The first one is, if we're talking about a small artery, small arteries or veins have little spiral muscles around them, okay? And woe be unto you if you cut a hole in a great big vessel, because you may have bought the farm. But if it's a little one, these little spiral muscles constrict immediately. It's part of the pain response. And so if you plug off the blood in that teensy little vessel, that lessens the chance of losing it out the hole. So we have that. Step 2 is the platelets, sensing a rough surface, secrete agents which cause them to clump together. Platelet aggregating factors, they're called. And that causes a bunch of them to stick together and form a platelet plug. But it's got to be a pretty small hole if that's sufficient to block the hole. Finally, the formation of a blood clot is the third one, and by far the most elaborate process. A bunch of really involved, very, very well understood chemistry is involved with that, and we'll talk about that. Okay, so vascular spasm is caused by pain receptors, okay, and arterioles are the smallest arteries, and that's where this is particularly effective. Arterioles lead into capillaries. So arteries carry blood away from the heart, and they branch out and bifurcate and get smaller and smaller and smaller. And pretty soon you get into the smallest arteries, which we call arterioles, and they finally empty into capillaries. Capillaries don't have any muscle, but arterioles do, and they're capable of almost completely cutting off blood supply to a region if all the arterioles shut down, as long as you haven't, damaged some great big artery. Okay. Vascular spasm is induced by local pain receptors. Okay, so this isn't a very good picture, but it shows those spiral muscles. That's one I got, clamping down on the arteriole, and it narrows the lumen of the vessel. And lumen is a word. What does lumen mean? It's a hole that the liquid flows through. Okay, it's the hole in a tube, and so that makes the lumen of the vessel much smaller. Okay, so then we have step 2. The platelets come in contact with collagen fibers, because if you damage the wall of a vessel, collagen fibers are going to start sticking out, and that really pumps up these little platelets, and they get all bent out of shape and start secreting all these chemicals. And many of the chemicals cause the platelets to clump together. Okay, and so here we show a bunch of them starting to clump together. Platelets are fussy little cells which are easily agitated, and they have the ability to release numerous clotting factors. And if they run into a not smooth surface, they'll start releasing all kinds of very potent chemicals. A series of reactions occurs to arrest the flow from the damaged vessel. When vascular endothelium is, so I say the same thing about 5 different ways. When this occurs, platelets swell and form spike processes. They become sticky and adhere to the exposed collagen. And they degranulate and secrete chemicals, real potent chemicals. Okay, and one that interests me a lot is thromboxane, which is a platelet aggregating factor. It is one we'll talk about. It's essential in the blood clotting process. Okay, this thromboxane, prostaglandin that's released by platelets, and this enhances both the vasospasm and platelet aggregation, as well as it's involved in certain clotting steps. And the inability to produce thromboxane compromises one's ability to clot. That's why aspirin, which is a prostaglandin inhibitor, functions as a mild anticoagulant, something we've mentioned before. Because if you block prostaglandin synthesis, you block the synthesis of thromboxane, because it's a prostaglandin. Okay, so formation of a platelet plug, well, if it's a very small vessel, that may work. But finally, we get to what we usually have to fall back on, the formation of a fibrin clot, which is an elaborate polymerization process. What is a polymerization? What is a polymer? It's a great big long molecule made up of a whole bunch of little molecules which you somehow trick into hooking together to each other. You take a whole bunch of monomers and do something to them to make them hook together like a choo-choo train. Okay? And they're all, that's industrial chemistry is fraught with polymerization reactions and finding catalysts that will induce various monomers to polymerize, to form polymers. And so we're looking here at the formation of a polymer called fibrin. It's a sticky polymer with a consistency kind of like it's a protein, and it has a consistency kind of like spider web. And things tend to stick to spider web, don't they? And so if you form a network of fibrin around the vascular injury, all kinds of junk will get caught in it, and that's what forms the fibrin clot. Okay? So you have to have a monomer which you cause to polymerize. Okay? There are two different pathways for the formation of this fibrin. One will happen just with blood all by itself. You could take a tube of blood and sit it out in a test tube rack, and after a while, probably a couple, 3 hours, it will slowly solidify and form, this will happen in there. And to do that, it utilizes the so-called intrinsic pathway. Intrinsic means it can do it all by itself. Okay? Extrinsic means it needs some help from the outside. Okay? And extrinsic pathway always involves tissue. But in the absence of tissue, blood will clot by itself. Okay? So the two pathways always happen in real life. They're always happening together. And so the bottom line, the overview means what's the bottom line? The bottom line is we got this monomer, and OGEN, have I mentioned this before? Maybe I have. If you see an OGEN at the end of a word, it means it's in its inactive form. So whole blood, whole unclotted blood contains fibrinogen, and that is the monomer. Fibrinogen is made in the liver. It's a plasma protein. It's water soluble, and it's one of the plasma proteins we find in whole blood. Okay? When the appropriate catalyst converts it to fibrin, the sticky fibrin traps various blood cells and forms the clot. So this becomes a discussion about how do you polymerize fibrinogen to fibrin? So remember, that's the ultimate goal, is forming a catalyst which polymerizes the fibrinogen to fibrin. And that's what this says. The story of clot formation is simply a discussion of how the various catalysts are formed. Okay? And so in the intrinsic, well, you'll see how this all fits together. But when you think intrinsic mechanism, the pivotal substance is probably PF3, which means platelet factor 3. It's a chemical called, it eludes me right now. It'll come to me in a minute. Platelet factor 3. Okay, so there's the intrinsic pathway, and it involves all these different clotting factors, and I think you can see that if you're missing any one of them or don't have the right amount of any one of these, that this whole thing is going to happen too fast or too slow. And maybe you've heard of having your prothrombin time done. If your prothrombin time is too fast, it means your blood is clotting too fast. You probably have too much of one of these things. Okay? That's why it's so important to have the appropriate amount of calcium in your blood. If you have too much calcium in the blood, your blood will clot too fast. If you have too little, it'll clot too slow. So if you have too much in your blood, the excess gets put in your bone. If you have too little in your blood, it gets taken out of the bone. Because it doesn't matter whether your bone density goes to heck. If you don't have the right amount of blood calcium, you'll have hemophilia. And, that's not a good thing. So here's PF3 over here with the intrinsic pathway, okay? But the part I want you to know, you don't have to know all this stuff, other than the fact PF3 is produced by platelets over here. But the ultimate goal is to make prothrombin activator. So here's the bottom line that you need to know. Both pathways produce prothrombin activator, okay? And there's a substance in our blood called prothrombin. We have two clotting proteins, inactive clotting proteins. If you put a pro in front of it, it means also that it's inactive. Okay? And thrombinogen maybe doesn't sound as good as prothrombin. So we have prothrombin and fibrinogen are both in the blood, in whole unclotted blood. And that's the only one of all these things that are in your blood when it's not clotted, okay? And so, when one of these pathways happens, finally prothrombin is created. Prothrombin catalyzes the conversion of prothrombin to thrombin, and thrombin catalyzes the conversion of fibrinogen to fibrin, and fibrin is the clot. So the goal is to produce prothrombin activator, which can be done either this way. There's a lot less steps in this process, so it happens faster. Damage to tissue is what causes this process to happen, and the damaged cells spew forth thromboplastin, also known as tissue factor, which causes clots, and calcium is required. And finally, you go through all these steps and produce prothrombin activator. This involves more steps, but it'll still happen. And you get prothrombin activator, and the catalyst is always written over the arrow because it is not itself chemically changed. And we have lots of processes in our body that are made to happen by catalysts, like enzymes are catalysts. And they're always written over the arrow because they are not themselves chemically changed. They just cause a process to happen. Okay? So this is the bottom line right there. So this is the extrinsic process, and we get this thromboplastin, which is spewed forth from damaged cells and results in the making of prothrombin activator. Prothrombin activator catalyzes the conversion of prothrombin to thrombin. And the thrombin, maybe you've heard of thrombin. It's often used as a topical coagulant out in the field in wartime or whatever. And thrombin, but there are no systemic coagulants. You don't want to inject thrombin into someone or they'll turn into a big clot. But thrombin catalyzes the conversion of fibrinogen to fibrin. And if that sounds like a bunch of Greek, by the next quiz, you should be able to say that, just like I'm sitting here saying. It's not the, once you get used to it, it's okay. So when are we taking the quiz on this?
Wednesday.
Wednesday, good response. You're still with me. Okay, so the intrinsic pathway involves PF3 and leads to the conversion of prothrombin activator. The extrinsic pathway involves thromboplastin and leads to the production of prothrombin activator. Once we get that, the same thing happens. Okay. Thromboplastin helps initiate the extrinsic pathway, while PF3 is the pivotal agent in the intrinsic. The two pathways meet with the formation of prothrombin activator. Prothrombin activator catalyzes the conversion of prothrombin to thrombin. Prothrombin is always in the blood. This is not always in the blood. Okay? It's only if your body wants you to clot that this is in the blood. And this is always in the blood, and when this sees this whole bunch of these hook together to form thrombin, which is a polymer. Okay? Thrombin, in turn, catalyzes the conversion of fibrinogen to fibrin. Fibrinogen is always in the blood. It's an inactive protein. It's this that's not always in the blood. And when the fibrinogen sees thrombin, the thrombin causes it to polymerize just like a bunch of cars and forming a choo-choo train. And this is the great big long protein, insoluble, sticky, spider-webby protein that adheres, forms a network around the vascular injury, ensnaring red blood cells and platelets and all kinds of stuff like that. Is a great picture. See all the fibrin strands and all the platelets and blood cells caught in there. Great picture of a clot. That's not so good. It's okay. So, injury in lining causes collagen fibers to stick out, and we form a platelet plug, and if that's not enough, we form a fibrin clot. And either PF3 if it's intrinsic, or thromboplastin if it's extrinsic, cause the formation of prothrombin.